What's going on, everybody? It is time for episode 8 of Half-Scaped. We have the Florida Scape Team here. If I can, there we go. Oh, there's Shelby. How you doing, Shelby? We have Brian down there. And there's Grant. How you doing, Grant? And
I'm all right, man.
Our special guest tonight is Kenneth. He is down in the bottom left there. I couldn't figure out which button to click to leave him off the screen, so we got an early introduction. So, yeah, how's everybody doing tonight?
Awesome.
Good.
I'm good.
I'm good now that I'm situated. How are you, man?
Good that you're situated now?
Yeah.
I'm good.
Sorry.
Good. It's all right.
I flooded a tank before I could get on stream, so.
You flooded it. Well, at least you caught it.
And it was on the 4th row, so it dripped down, and yeah, Shelby caught it.
Well, that's good. That's good.
At least it was early.
Yeah. It wasn't like to where it's flooding multiple tanks at the same time. Like,
no, it was, but
it
didn't make a puddle on the floor yet.
All right, perfect. Do you purposely leave the ones on the bottom a little bit lower just for that?
No.
That's why
No,
he has Shelby.
on the lower level, the 20s, they're a little lower just because like the metal started to bow a little bit with them filled all the way up. So I slacked like 2 inches off for the 20s.
Nice. Well, I guess we'll skip all the questions and how's everybody week. Like we'll do what we did last week on your channel. We'll just kind of jump right into the topic because we tend to get like long-winded. So, so today's topic is going to be bacteria in your aquarium, and basically we brought on, in my opinion, probably the most intelligent person in the field of bacteria in the hobby that I know of, that I've spoke to personally. I've had it driving home one time from a trip. I spoke with Kenneth on the phone, and he blew my mind. So the man's a wealth of knowledge. So if you guys are, curious about the roles of what's happening in your aquarium and stuff like that, this man here more than likely knows why and how. But Kenneth, tonight's topic, like we were talking about before, is bacteria in the aquarium. And I know that you tell us a little bit about you. Tell us a little bit about like your company and what you do and what you've developed.
Sure. I've been an aquarist, I guess, since Ronald Reagan was president. So a long time. I think the longest I haven't had a tank was when I lived on a fishing boat. So it's just kind of always permeated my life. I've worked in the industry about 30 years, almost continuously, with some side stuff like plant nurseries and working in vineyards and so forth. But years ago, while I was in school, I worked at a lab breeding. Well, we were breeding fish, but it was a zebrafish lab. But my main responsibility was cultivating the live foods. And that included like paramecia, stuff like that. So micro stuff. Nice. And yeah, so it just kind of, became my chosen specialty. It was a lot more interesting than I thought it was going to be. Kind of ventured from, again, I guess I kind of got into smaller and smaller stuff as time went on, because then I started moving into like phytoplankton and all that. Actually, I moved here to Colorado to work for, I came out to Colorado to jump into the, at that time, emerging cannabis industry, and ended up, I guess you'd say, take a U-turn, but I ended up doing what I kind of usually do, and started working for Algae Barn.
Awesome.
Yeah. When they were, at that time, they were in a little garage here in Longmont. Well, it was a little garage, but it was a garage. It was nothing compared to what they're doing now. They have an actual campus with multiple huge buildings and everything. It's pretty ridiculous. But back then they were doing nothing but pods and phyto. And actually, before I came down, because I was developing products for them, I was basically their process biologist when they were getting started. Well, they weren't started; they were already going, but they wanted to expand into other things. And one of the things I wanted to bring down here and implement was a cultivation program just for these bacteria. And the intention originally was just to grow them in house for the purpose of cleaning up their copepod cultures, basically remediating the water, because when you grow, copepods, you raise them, and when you do it the way they do it, they're in really high density, so the water fouls quickly, and also they feed them pretty intensely, and they're usually aiming for like a carotenoid-rich food, particularly for the Tigriopus, because they want them to be that nice red color. Well, in looking around, I found these bacteria. I'm like, well, hell, you could do both things. We can clean up the water, and they're rich as hell in carotenoids, including astaxanthin, which is literally worth its weight in gold. So I'm like, wow, this thing's actually going to
dual purpose, 2 birds with 1 stone. Nice.
Yeah, it's like they cleans up the tank, and then as they proliferate, they just become an uber nutritious food for these copepods, and then they even color them up. So It's just a win-win-win, right? But when I came down, so Algae Barn, I know it's mostly freshwater people here today, but for whoever's familiar with Algae Barn, they do all kinds of stuff now. At that time, they were just doing pods and phyto, and we were expanding it at that time into like macroalgae and all these things. It just got wild, and this project that I was just talking about got put on the back burner. And anyway, when they expanded to the point that they had to move and they went closer to Denver, I stayed behind here and started the company Hydrospace, just to cultivate these bacteria. I thought they were so important that it wasn't a matter. I wasn't even considering like doing phytoplankton or any other kind of bacteria. I just wanted to focus on purple non-sulfur bacteria.
And a lot of people overlook it. Like a lot of people don't realize the importance of the bacteria in their aquarium. Like, I talk to people daily who are, asking questions about algae growing in their aquarium. And their tanks are like 2-3 months old, so like super immature, and they've already nuked it like 5 times at peroxide. So it's like, they're their own worst enemy in a way. So a lot of people overlook what you do and what it is that you cultivate, and the importance of it for a stable, healthy tank, even on the freshwater side. Like it's more important than, I think, fertilizer at any rate.
Well, yeah. So I'll jump in here real quick and just say, like, hey, Ken.
Hey, bud.
Anyways, so somebody that does a biomedia and stuff like that, and talks about managing your biomedia and the biofilms. I've known Ken for a long time. I tell people he's the first one since my wife, or the first person since my wife before I got married, that kept me up all night talking on the phone. So we've had some good conversations over the years. But one of the cool things that you I don't know if you touched on yet is it's also like a probiotic. So like your bacteria is actually meant to be dosed. It could be added to food or dosed periodically to kind of be like one of those maintainer of the clarity, balance, nitrogen fixation, and all that stuff. But my real question is explain to us, how you use aquatic plants to culture your bacteria. And, I think one of the coolest things about the way you culture it is it's already biologically available. So when you dose it in your system, it's already something that's already in your environment. You're just enhancing it. So I'll let you go ahead and explain the plant side of it.
I think you're referring to how we make it. The culture medium. Yeah. So that's one of the products. PNS Pro Bio is cultivated on an herbal extract. It's made from Myriophyllum spicatum. Some of the plant people on here are gonna know it's a highly invasive plant. It's from Eurasia. What's that?
Super invasive.
Yeah, and really destructive. So it's really hard to get a permit to collect it or anything like that. Like especially around here, Boulder County, it's especially invasive. And we have like decontamination centers around a lot of lakes and stuff where they pull it off of people's boat props and so forth. It can dry out and survive and be carried into another area. So it's a pretty big deal, and it's really bad. It's up there with like
Pistillas, the
zebra
water
mussels.
Yeah,
What's that?
yeah. The pistillas down here, the water hyacinth and pistillas water lettuce as well. Like it's the same deal. You could have a little piece on your prop, and like 3 months later, drop your boat in a different body of water, and then that whole lake's infested.
Yeah, I think they call them like the ghost plant or something like, or maybe zombie plant. They call them because they're able to, they seem they're dead and dried up. They dry to nothing. But anyway, so it's hard to get a permit to collect something like that because it's so highly controlled, and natural resource managers are scared shitless about it moving around. So I really had to go the extra mile to convince them to let me harvest it. And ultimately, it was the city of Boulder granted us a permit, 2 different sites. This year, I'm waiting for that permit. It would be our 6th year. But what we do is we just go in, waders and all, and battle leeches, snapping turtles, and whatever else.
All the fun stuff.
What's that?
Yeah.
All the fun stuff.
All the fun stuff. It's actually pretty fun. It's interesting to get out in the field and see, get into the natural environment, get a sunburn, all that. But we harvest about a half a ton a year, and that dries down to much less than that. But basically, we put it out we de-water it, as they call it. You just basically put it out in the sun and dry it as much as you can. And
what does that smell like? Sorry.
It smells, believe it or not, a lot like tea. And one reason for that, it's super rich in polyphenols. So Myriophyllum, one of the reasons it's so invasive, and also one of the reasons it works well for this bacteria, is that it's rich in a tannin called telemigrandin 2. And it mostly kills cyanobacteria. Not epiphytes, planktonic. But the reason is that they kill all the plankton in the water, and then they have more access to the sunlight.
Right.
So they're actually not very effective against epiphytes, but they can clear the water column real easily with this stuff. It's highly algacidal. So mostly effective against cyanobacteria, but it'll kill some green algae as well. And I guess it disrupts. Now, this is where I'm starting. I'd have to get back into the books to say for sure. I think it disrupts photosystem too. And photosystem, now these bacteria that I cultivate are pretty ancient. They're actually the ancestors of all cyanobacteria and therefore green algae and therefore all land plants. But they're more primitive. They don't have a photosystem too, so that doesn't affect them. And in fact, they can eat the stuff because these particular bacteria can degrade those types of they say recalcitrant organics that aren't really broken down easily. So I don't want to dwell too much of that and put people to sleep, but there's a reason lignin and cellulose are so abundant they're the most abundant organic substance on Earth, and that's because they don't break down very easily. So these bacteria are kind of among the few that can degrade cellulose, and that's why you see them in pond products. They'll call them sludge digesters. They'll eat like leaf litter and stuff like that in ponds. So they do the same thing in a planted aquarium, but they can use this stuff as a carbon source. So anyway, that's what we do with ProBio. We use this tea. It's kind of has a positive ecological impact, a net positive impact on the environment, which is kind of cool. And right out of the bottle, it's already adapted to consume, because they do. So the bacteria behave differently based on how you cultivate them. So doing it this way, they're actually out of the bottle adapted to consuming those tough organics. So in a planted aquarium tank, where you have all that junk on the bottom, they're kind of useful in just clearing up detritus and all that.
So yeah, I was gonna say, like, almost like a prophylactic for algae in an aquarium, like just to, because obviously in low enough, amounts, this, wouldn't affect hard cellulose, like, the plants that have, healthy growth on them.
Mhm.
So that would be a really good, resource of a prophylactic for against algae. As would be like a small dosing regimen of this in a planted aquarium or any aquarium.
Yeah, more so because they compete for nutrients with
Right.
your so-called bad algae, so and space. But they do most. So one thing about these bacteria, I should probably describe the bacteria a bit. They're phototrophs, so they're photosynthetic. Like I said, incredibly ancient. They're really tiny, of course, being bacteria. They're only like a couple millimeters, micrometers long. So you can barely see them under a scope.
So
And
yeah, so that would play well like in an aquarium where people are lacking. Like the biggest thing I deal with is people who don't understand plant mass and the deal with plant mass and how it takes up the food for the algae basically. So
Mhm.
I mean, for those folks that are trying to run these like extremely hardscape filled tanks and such where they can't have plant mass, this bacteria would, it's like the answer almost, a
Yeah, it would just
micro plant mass.
yeah, it'll occupy that niche and
Right.
consume nutrients from that, like the anaerobic zone.
Right.
They're primarily anaerobic. So these are a lot like in fact, they're closely related to the bacteria that are associated with legumes. They're in the family Brady. Well, actually, they're going to reassign them, I think, but they were in traditionally they're in Brady Rhizobiaceae. So they're in with like close to like rhizobia and stuff like that. Brady rhizobia. These are the ones that so like you hear about cover crops, right? So um, uh, clover, alfalfa, stuff like that. The bacteria invade the plant's roots, and then the plant will respond by creating a nodule that the bacteria can live inside of. And for the most, aside from just the physical protection, it creates an anaerobic area. And that's important because nitrogen fixation can only occur in
An anaerobic
the
environment.
absence of oxygen. So basically what these plants or these bacteria do is they take nitrogen gas out of the air and they turn it into ammonia. So basically they fertilize the plant from within, and then the plant secretes in a very targeted and deliberate way. If plants can be deliberate, they release organic substances to the bacteria to encourage them to grow. So it
So
most
fully
definitely is
symbiotic.
fully symbiotic. Now with like rhizobia and the ones that are associated with legumes, those are obligate symbiotes, I believe, but it's an endosymbiosis. The bacteria live inside of the plant. These guys are more generalistic. They'll live on the outside of the plant, and they don't depend on the plant for growth. They can live. They've been deep sea sediments, forest soils. They've been isolated from the human belly button, so they can live anywhere. And but they really like plant roots for that reason. And because they're generalists, they'll live on all kinds of plants. So you see these products being used for everything from pond plants to cannabis.
Yeah, like a mycorrhizal bacterial, additive and such. That's what I've been applying yours as lately for the last while, as well as a foliar. It's differences there, definitely.
Yeah, so both have been studied. The foliar applications, even on terrestrial plants, the bacteria have been shown to survive the drying because they're not spore forming. They can't survive drying out. But what they do is they invade the tissue of the leaf, like get in between the cells or something. These
And
little
the
nooks and
stomas,
crannies.
yeah.
I think I've seen even just between, just kind of all over the leaf. Any little crevice where they can avoid drying out. There's a die-off, but then the few that survive will start to grow back again. And I think because when you see how they grow back in like tight groups, so they must conserve water inside of a little biofilm or something. But
well, in a paludarium application where the humidity is super saturated as well, I mean, there's probably extremely less die-off too, and that's it's kind of what I've been noticing is the things that I'm using or treating, however you want to put it, that are extremely humid environment are so lush, much more so than the ones that aren't being treated with such. So it's cool. It's cool to see, you know, the difference actually side by side.
Some of the stuff like that you'd see in a paludarium or a vivarium, like bromeliads, a lot of like those epiphytes, like plants that don't necessarily have a root system, they really benefit from these bacteria. And I've known people that have greenhouses. A guy that does orchids, and he said the minute he was doing a foliar application, and he said the minute he started doing it, some of his stock that had never bloomed before just started blooming like crazy.
I'm sure. I mean, the aeroids, the tree climbing aeroids and such, that rely heavily on aerial roots, it's the same deal. Like all my philodendron and stuff that I'm treating with this are like, they go nuts, and it's all because of the symbiotic relationship of the bacteria feeding the plant and the plant feeding the bacteria back and forth. I'm barely having to even use applications of fertilizer in some cases, you know, like
I'll jump in real quick. So the way I usually dumb this down, like it shows, I usually like look at them and I say bacteria is lazy, just like me. It only wants to eat and reproduce. But it's the same thing for plants and everything up the food chain. Like, you know, your microorganisms want to eat bacteria that their bellies are full and are mass produced. You know, it goes all the way up the chain. The same with the symbiotic relationships with the plants, where they can survive in the leaves because they're providing benefits for the plants. So, yeah, it makes things grow easier when they can they have what they need to grow more effectively. It doesn't take like actual physical energy that they have to produce to do that, where, you know, something else can do the work for them.
Right. We're going to jump to the chat for just a second because there's like a lot of people asking questions, and I don't have any mods or anything, so I've been zoning out talking to you and completely forgot about all of that, so I apologize. So
also,
real quick,
shared screen.
No, I cannot. There's no shared screen.
Let me try to share one with you so you can.
All right.
It might help. Ken can explain this really well. It's an old high school experiment. Let's see.
So what's going on, Omid? How you doing, Ryan? Dark River Aquatics, what's up? Here's a question for you, Ken. I've been trying to get approved for bringing some invasives from the wild into home aquarium. Curly pondweed and Potamogeton crispus is the one I really want to get approved for the most. I guess he's asking do you have any experience with dealing with getting permits? From my experience, I guess it's varies state by state. Florida is completely different than most states. So.
It might totally depend on who you talk to. I mean, I've had people tell me flat out, no, I certainly wasn't approved the first agency I approached. Right. So, and obviously that curly pondweed's pretty invasive stuff. Yeah. So, I mean, you might have to, you know, I just say, go for it and just tell them what you want to do and make it clear that number one, you have absolutely no intention of cultivating it outside of the home or transferring it in any way. But also they might, if you're harvesting it in a moving body of water, like a creek or a river system or something, you might want to propose using a net to capture debris, because that's the principal way a lot of those plants are distributed, is through fragmentation like that. So anyway, yeah, just adding that net thing in there might just kind of demonstrate that you've thought this through and you're extra careful about preventing further invasion just from your activities.
Exactly. Yeah, you don't want to add to what is out there. um, here's another one. Are you saying plants don't consume nitrogen? That bacteria breaks down nitrogen to ammonia for the plants to be used, or did I misunderstand that?
Yeah, maybe I wasn't clear enough, but plants, most certainly use nitrogen. Of course, they have to. It has to be fixed nitrogen, meaning they can't just take. Our atmosphere is 80% dinitrogen, just nitrogen gas, but it's completely inert. Even plants can't use that. In fact, there are hardly any life forms on Earth that can use that. It has to be fixed. So what they mean by fixed is it's converted from that nitrogen gas into ammonia. So these bacteria have the enzymes that are required to do that. Sometimes they're called diazotrophs, but nitrogen fixing bacteria is easier to remember and a little more self-explanatory. But basically what they do is they introduce new nitrogen into an ecosystem. The only other means of that, I guess, like lightning can do that, but that's a piss poor way of adding, when you have all the nitrogen leaving an ecosystem from denitrification. Just, assimilation as things grow. So anyway, these bacteria introduce new biologically available nitrogen into an ecosystem. And that's why they're important to a lot of horticulturists. So they'll add it to either hydroponic water or pond water or even soil, and the bacteria find their way to the roots and then start that association with the plant, where the plant feeds the bacteria by secreting organic substances that the bacteria can eat, and then in return the bacteria, there you go, and the bacteria fix nitrogen. So they literally just make ammonia out of thin air. So they're fertilizing the plant right at the root, if that makes sense.
Yeah, so Ken, the Winogradsky column, you explained this probably best to me, and I think when it comes to the plant world, this will kind of help people, because this is basically in high school yeah, there we go. Yeah, it's a high school experiment where you go and take some pond water and you can watch the colors of the bacteria basically adapt to the column. So yeah, go ahead, Ken. I'll let you explain it, could you explain like the cyanobacteria and kind of like the levels of the breakdown? And I've always kind of imagined the purple non-sulfur as like the TIE fighters that'll kind of mess with the layers and balance out the layers of your substrate.
They're kind of in the middle, yeah. I wish I'd done this in high school. My first Winogradsky column we made in my college microbiology lab. But basically what you do is you take a bunch of sediment from like a river or something like that, and you need a source of carbon. And what people use sometimes just paper. Cellulose, you'll have bacteria that can actually, like I was saying, there are some bacteria that can break down cellulose. And that just kickstarts the whole thing, usually from the bottom up. So, and this, you see the gradients, right? On the far left, there's sulfide, and that is heavy towards the bottom. It moves up. So, and then oxygen, of course, as you go deeper into the mud, oxygen is going to become depleted as it's used by aerobes at the surface. So anyway, what will eventually happen is these different colored bands will emerge inside the column, as you'll have really high densities of bacteria in these microzones, or it's called lateral zonation. But yeah, so at the bottom, the sulfate, well, let's start from the top. Obviously, cyanobacteria and green bacteria, whatever, green algae, any kind of aerobic phototroph can survive on the surface where there's oxygen. And then below that,
typically the ones that people are battling in their aquarium constantly.
Yeah,
definitely. Yeah. Yeah. And then heterotrophic bacteria is all kinds of stuff, everything from like bacillus and in that same area, you're going to have like your nitrifying bacteria too. They're not heterotrophic, but they are aerobic, and they'll be up on top like that. And then the iron oxidizers, obviously right under that, you can see they're microaerobic. So they can tolerate some oxygen, which makes them a little more competitive against the anaerobes. And at the surface, they still have plenty of access to iron, which they use in their metabolism. I'm honestly not as familiar with those bacteria. I just haven't needed to look at them much. And then below are the ones I know pretty well, the purple non-sulfurs. So you can judge by the names, purple non-sulfur and purple sulfur bacteria, that the purple sulfur were probably discovered first. And then right? So the purple sulfurs, they actually use hydrogen sulfide in their metabolism. So they oxidize it, and it's part of their respiration. Anyway, they're actually more similar to the green sulfur bacteria. Really, the big difference between the two is just the type of pigments they produce. Those guys are obligately anaerobic, meaning oxygen will kill them. Purple non-sulfur bacteria are they prefer anaerobic areas, but they'll definitely survive in an aerobic area, and that's why you see them up above that. Also, the other reason too is that hydrogen sulfide is somewhat toxic to purple non-sulfur bacteria, which is why they got their lame ass name after the fact. Right, just because the purple sulfur bacteria were discovered first. But basically the sulfate reducing bacteria at the bottom, those guys are the ones, like if you've ever walked on a muddy creek and your foot sinks into the mud way down into the anaerobic stuff, and you hit that black stuff, the hydrogen sulfide that smells like rotten eggs, it's that hydrogen sulfide is made by the sulfate reducing bacteria. So hydrogen sulfide is toxic, and it'll kind of come up through the substrate and but it gets used up along the way by the purple and green sulfur bacteria. So it's not much of a problem by the time it hits the purple non-sulfur bacteria. So I probably hit that enough, but
let's say you're in a Planet Aquarium and you are taking your hands and pulling plants out and doing maintenance, and you disturb those zones, obviously then you're going to have some issues, correct?
Yeah, and that's why a lot of people, there are a lot more proponents of like a no-till agricultural systems, and stuff like that now, because there is that stratification of microbial communities that occurs, and it's not an arbitrary. They pass nutrients and oxygen and things like that from in carbon to one another in its appropriate form. So you disrupt that, you break it all apart, and then it kind of has to reassemble itself. But it does. Correct. So if you're just pulling a plant out here and there, it's not a big deal. If you go through and just completely demolish an aquascape or a field or whatever, it's going to take a lot more time to recover.
So it's been 30 minutes. Can I say something about the topic now? Hi, I'm Grant. We haven't been introduced, Ken. I mainly keep shrimp. I got like 300 shrimp tanks over the house with Shelby. Shelby went to go feed her fish. But one of the big issues with keeping shrimp is when people mess with a plant. It doesn't matter how many plants or what. It just seems every time somebody grabs a plant out of their shrimp tank, it causes some type of bacterial infection. And now I'm wondering, maybe it's the sulfur reducing bacteria that are causing this.
Are you smelling like rotten egg smell, or seeing like black stuff come up from the substrate, or even on the roots of the plant?
Usually.
That could be then.
In shrimp, almost everybody's using the aqua soil. So when you rip it up, you've got that black sludge regardless. But yeah, it just seems to be an issue. I know you said it doesn't really become a problem with too many plants, but it seems with shrimp, they're invertebrates, maybe a little bit more sensitive. It's definitely an issue in our hobby.
Yeah.
Yeah, it could be so many things, because even disrupting that and introducing new chemicals or new bacteria into the water column will just completely, even like on the phylosphere and in the water column, everywhere, it could change that and affect the animals in some way.
I was just more curious with your background.
And shrimp, I think, ingest.
Like that, if you knew the sulfur reducing bacteria, if it was, I don't know, poisonous to inverts or something like that.
Not the bacteria, but if so, you see this in reef tanks a lot, where people have real deep sand beds, and you get a lot of organics that get to the bottom, and with sulfur, containing organics that get metabolized by those bacteria, and you get that black layer underneath. And sometimes when people dig into that, or even a fish will dig into it or something, any burrowing animal can disturb those layers and bring the hydrogen sulfide up. The hydrogen sulfide is toxic, yes. And for a sensitive animal,
now the next thing is everyone really desires, the algae growing on the walls. There's two different kinds usually that grows. You get the green, and what's more preferred is the brown algae. Is the brown one that grows on the side of the freshwater tanks, is that the purple non-sulfur bacteria?
Likely not. It's probably actually an algae. Yeah, like a diatom or something, if it's a golden brown color. You're going to see, if you see these bacteria, it'll be against the tank, like underneath the sand bed. And when I say purple, they're usually quite purple. So purple non-sulfur bacteria, like Rhodopseudomonas, can kind of be a rusty color, but it goes from that rusty red to burgundy to like bright purple. So it's at least some shade of purple.
Super rusty on the substrate, especially with some of our older tanks. We held them out in the garage. I'm looking at one right now. It's super rusty and orange color, and it's probably from the top of the substrate to an inch down.
Oh, from the top? Yeah. So it could be purple non-sulfur bacteria. Some are, because the carotenoids, right? Like carrot, the carotenoids are usually pretty orange colored, but again, too, that may literally be rust from the iron oxidizing bacteria that would live closer to the surface.
Okay.
So
and that would precipitate down, right?
It could, if it gets to it could get into the anaerobic stuff and be more soluble again. And
is the iron one good?
Yeah, I mean, they're all good in their own way, because they perform these. They each just play a role in a larger function that cycles phosphorus, nitrogen, carbon. So yeah, they're all good. They all kind of have to be there. That's why it's like they talk about microbial communities as being guilds, right? Where they're like, you have all these different pieces of a puzzle that is one picture, and that picture may be the microbial community of like a shallow creek sediment, or for a flooded forest puddle or something. Each community is unique, but from other communities, but each community looks pretty similar because each piece has to be in it to function ecologically.
So I got a good question here, and it kind of ties in with my own little personal question. So someone's asking if this type of organization will happen in a 3 to 4 inch aqua soil tank. I think we pretty much just covered that. Yeah, it will, right? But question, I guess, really on top of that question is about how long would you say these cultures take to fully develop, and this ecosystem to fully, become symbiotic?
It could take years. It depends. So first of all, the bacteria have to be present. So the tank needs one way or another to get inoculated with those bacteria. And you'd think that the saying is life finds a way, right? You'd think just by moving a plant from one tank to another, something like that. But that isn't necessarily so. I mean, you think about how we trade plants often, they're just damp. They're not even immersed in water, right? In some cases. So obviously that eliminates half the bacteria in this column here, just because they're obligate anaerobes. You kill them right off the bat. So sometimes it's if say you go out into a creek and you scoop a hand of that muddy, nasty sludge or whatever, and throw a little bit of that might speed up the process, especially if you are able to maintain the anaerobic conditions in the core sample you draw and kind of insert that. I know it sounds like a pain in the ass, but that would theoretically be the fastest way to inoculate a sand.
If I do half the core sample as I draw, you're gonna wind up with like Gibba and all sorts of other nonsense too. Because, like there's so much Utricularia and bladderworts out where I take samples.
Mm-hmm.
Yeah. So the reason I wanted to show that slide and have Ken talk about it was because, to relate it back to the aquarium in your system, the reason why you always hear, you have to clean off the detritus on top of your substrate. And that's important during your water changes, because your substrate's not meant to be your filtration. So if it's got that layer above it, just like Ken said earlier, like his bacteria will fight against the cyano layer just to get the light on it. But if you constantly don't have any of that interaction happening, that's part of the reason why people find that if they keep their substrate clean, they have less algae issues, because their water quality, they're using their filtration as a way of breaking down their organics versus trying to make your substrate to use it, especially when you only have, a limited space within it. But going back to the Winogradsky column, so I've always found that Ken's bacteria, especially when I use it in a planted tank or a plant aspect, like I said, it kind of acts as that balancing point. So you see a lot more of those issues, or it's harder to cause those major issues when you do disturb it. But again, I'm a big proponent of dosing it regularly, whether it's with your food or, yeah.
So another good one, anything in specific substrate wise that you would recommend that these bacteria would build up best in?
Clay soil, specific depth substrate. In a substrate, it would depend on the size of the grain, but generally I found about an inch below is where you see it growing, roughly, maybe a little less than an inch. And then the band is usually about an inch wide. But so that could take longer, and then it's subject to being disturbed, right? Anytime you're digging around in the tank or pond or whatever. So often some kind of biomedium is best to grow them in. And that could be, Brian just missed his chance for the plug here, but
exactly, Brian, you welcome.
But
no, I was actually looking at my tank. I have a bunch of your bacteria.
But so any kind of like highly porous material will work really well. Just so long as that you have that on the outside, you have all the aerobes that consume all the oxygen, and then deeper inside the core, these guys are more competitive. So like aquachar, would be a good example.
Yeah,
for the reason
so that's actually, to go into that, that's the reason why I switched out. If you watch the earlier episodes, I had the white sand substrate. I switched it out to the aquachar flakes, and I actually have some mesh screening I can put over on my as a layer between my substrate. So it would be something that you can kind of, when you're if you break the tank down and stuff and pull it apart, you can use it as a, we have a regeneration process and a way to clean it and stuff, but I won't go into that. But there's a way to use it as a substrate base and also use it as a jump starter for your next tank. Because like you said, as you're building the layers of the substrate, you also can build it within a porous media internally as a microsite.
So that's exactly what I've been doing. I've been using your aquachar and Ken's bacteria and having a blast. So the applications are endless, and in young tanks and fresh tanks, to where you don't have this development yet, to where your substrate isn't as colonized as you want it to be. Keeping that top layer clean is very important. Otherwise, things get clogged up, and you don't grow the right bacteria, or you grow the wrong amounts of the wrong types of bacteria. In my experience, anyways. But to have that biomedia available for the right types of bacteria to colonize on, away from where you constantly need to clean, so you can always have that bacteria available in your aquarium, is priceless. That's how you get these amazing looking planet aquariums. Is the Dutch guys know about bacteria. They know the importance of bacteria. It's like the most unspoken secret in the Dutch world is bacteria.
That's why the vials are popular, because
Exactly.
it's a constant dosing of bacteria. Because a lot of hobbyists tend to get in that mentality that once a tank is cycled, it's cycled,
And that's such a
and
myth.
it's not something that you should actually manage and mature. And I try to say, the investment into actually managing it, maturing it, and like I say, like treat aquachar like live rocks you can rinse when you're using it as a biomedia. That type of management of a system is super easy and cost effective. Like it makes your husbandry in a bunch of different aspects of life, that baseline knowledge, pretty interesting.
Yes, sir. Is there anything trying to catch up with chat? Sorry. I gotta figure out how to make mods.
I would just say that with the biochar, you might be interested in this too, Chance. It's actually a really favorable medium for these bacteria to grow on. I've seen studies where they compared biochar to sintered glass, which I suppose was used as like an inert material or whatever. But biochar has residual organics inside of it, and I wouldn't. Brian would know better specifically what those are, if they're sorts of tars or whatever. But apparently they're labile to these bacteria. They can eat them, and because of that additional food source. And by the way, it's one that most bacteria can't eat. Like I said, again, these bacteria can degrade these tough organics that other bacteria can't.
Yeah.
Specifically cyclical compounds and cellulose, lignin. These lignocellulosic compounds are kind of just burnt into the biochar, and it's like a food that's just kind of left there for them. And in the study I saw, I think it was 11%. It was an 11% increase in growth on biochar compared to sintered glass, which would be like I think they used Biomax from Hagen in that study. Yeah, but that would be like Cyperax or something like that too. Yeah, very similar.
Inert colonization, such.
Yeah. So the difference between a regular biochar and stuff in that studies and aquachars, we're actually engineered for aquariums, so we're meant to be that initial biological reaction. The agriculture biochar and stuff like that you'll see used in the hobby and stuff like that. It's can take a year or more to be able to become that biomedia or the microsite where you actually can build up that diversity. So the difference between the biochar and aquachar would be more from an aquatic standpoint. We do we're more of like that activated carbon, so we can decontaminate. So if you think of, even if with terrestrial plants, if you water your water, it's going to dissipate whatever contaminants it's in the water, or the water's going to dissipate it. It actually acts like a black hole that can form the biofilm over it and stuff like that. So I digress.
I got a question for Grant here. You mentioned that the bacteria when pulling up plant may cause shrimp issues. Anything to combat that you still have rooted plants in shrimp tank? Heavy water change same time maybe?
Yeah, heavy water change is really the only thing you can do about it. Basically what I've done is I've stopped touching any rooted plants in my shrimp tanks. That's why a lot of my breeding tanks are just moss, or I might have some like Booster Anubis growing on some wood, but I've stopped putting crypts in my shrimp tanks for that reason. So.
Crypts will do it.
So sorry, I was reading through.
And I would imagine that would be kind of like making like a spring or a well, where like if you pull up a plant root, especially like a crypt in the system, it's a long spidery root. It would almost seep that that sulfur or sulfuric.
I've definitely seen with crypts because the roots die off so bad.
Oh, yeah.
And you always see that hydrogen sulfide and smell it when you pull those. There's so much dead material underneath.
Yeah. Well, that's what I wanted to. Yeah, go ahead. Sorry.
I was gonna say you don't have to get rid of the crypts. Like we have a couple tanks where there's shrimp and crypts in it. Just I'll probably let the shrimp like run its course, and then when all the crypts have taken over the tank, I'll take the shrimp out, take the crypts out, and then reset the tank and like put one crypt back and let the course redo it. But the crypts will pay for the new substrate. So
I was going to
it's a good
say,
cycle to go for.
Grow the bushes.
That's what I was going to. That's what I was getting at. To come full circle, basically back to stability. If your goal in mind is to breed shrimp, set your tank up to breed shrimp. You know what I mean? Obviously, the goal is to most production of shrimp you can, which is Grant's expertise there. If your goal is to, or one of Grant's many expertises, if your goal is to, set up a successful like Dutch style planted tank, try not to move stuff around as much. Have a plan, lay out your scape, think ahead, do some research on the plants as how big they're going to grow. Where do you want them placement wise, blah, 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 blah. And plant mass, pack them full. And leave them alone. Try to stop from pulling things up and pulling things out all the time, and let your tank establish. Because like we were just saying, I mean, a lot of people when they first start this hobby, they sit here and they play gardener all week and, and keep moving stuff around and move this there and move this here, and then get upset when they have algae issues, and then they use algicides, chemical algicides. And it would be amazing if we could actually stop using antibiotics to treat cyano and start using regular, nice, bacteria that already should be in our aquarium colonized and eating it as it is. So just to come full circle, everything relies on stability, because these bacteria aren't going to grow in completely unstable conditions. They're not going to grow right in conditions that are continuously changing, unless I'm wrong. But
not from what I know. Yeah.
So Ken, I want to talk about the kit that you sent me, because you've got because right now your stuff's just a general. There's a couple different lines that are more of a general PNS. Like a probiotic is what you call the line and stuff like that. But I know Chance and I have been working on some of your different lines of bacteria outside the Palustris strain that you typically do, I think. So I want to go over the little kit that you sent me. Let me grab the...
While he's finding that, basically, quick question. Could you use a pipette or a syringe or something to inject Ken's bacteria into newer substrate to inoculate it? So, yes.
Theoretically, you could, and theoretically, it would work. I don't know of anybody who's actually done that, but it totally makes sense. If you could get it down into an anaerobic area. Now, these bacteria are not that sensitive to oxygen. They can actually function in oxygen. It's a different metabolism, but they'll survive as just regular chemoheterotrophs. For those that means anything. The aerobic bacteria that can just consume organic matter, just like you and I do when they're outside of that realm. But that would give them a good quick start. Certainly make them more competitive against other bacteria. And another advantage I can see to that is that you can concentrate them. And the reason that's good is that they're more competitive when they're densely, when they're in a high density. So basically they communicate with each other. It's called Quorum Sensing. And believe it or not, these bacteria can send messages to each other about the status of their environment and the status of their own selves. And when they're when they, for example, when they all know that they're together in a large number, large group, that signals that there are enough of them to where it would make sense secreting antibiotics. So say you have a new substrate and you want to like pipette it or syringe it into a deep area of that substrate. Presumably that's already anaerobic if it's not super new, but it would go anaerobic pretty quickly. You could eject them directly into there, and then the other bacteria that are almost bound to be there, it's hard to create an area that doesn't have any bacteria. So there will be some there that are existing, and they may compete with these guys. But by pipetting it in like that and having a whole bunch of them at once, you give these guys a competitive advantage, basically, because then if they decide through their communications to release streptomycin or kanamycin, they can then create what's called a zone of inhibition, where collectively, where they secrete all these antibiotics, they'll kill everything around them and it'll give them more space to grow in. So it's basically like chemical warfare. And if you're just an individual cell by yourself, it's not useful for you to waste the energy to release that antibiotic, because it's just going to get diluted and not hurt anything around you.
Now
So
you got me wanting to create a septic zone and inject it and see what happens.
yeah, but that's actually
a
a great idea. Honestly, if someone were willing to take the extra time to inject it, it would not only speed up the colonization, but increase the likelihood that the bacteria would persist there.
Nice.
Yeah, I like that. So nice, Ken. So tell us about the capsulatus and your grow pack, because I'm definitely going to be injecting it now that you mentioned that, because I mean, I'm a big fan of the substrates, and I'm working on the flakes on this tank. So tell us about your strain of plant bacteria and what makes it different.
So that's Rhodobacter. That's one of the 3 species we cultivate. That one is strictly freshwater. So it's never been really popular with us because most of our business is with reef people and so forth. But capsulatus is pretty badass. That one, I was just reading a study today where, as a diazotroph, a nitrogen fixer, they've actually added that species to hydroponic systems that had no fertilizer, no nitrogen anyway. Yeah. And it completely supported normal growth rates by itself just through nitrogen fixation.
Awesome.
So very powerful, very effective. It's not just something that might make a little bit of a difference. It makes all the difference.
Yeah, that's huge for like aerospace or space cultivation. Like, then they could just cultivate the bacteria in the space station and use that bacteria as the nitrogen fixation on the plants without them having to worry about liquid fertilizers or any of that jazz.
Could you hold that product back up, Brian?
What's that?
Could you hold the product back up? I'm sorry, Chance.
Okay.
You're right.
So this is the explain the starter kit. So it comes off with like a starter bottle, and then this is the 2.5 gallon starter kit. So this is like a plastic bag, but it's kind of like your milk pouches. You fill it up with RO water. He's got a seeding agent in here, and then there's a cap with a little spigot on there, so you can kind of take a little bit each day and put it with your food or top off water or whatever you want. And kind of the way I've always used Ken's bacteria has been always a daily doser almost. Even a little bit goes a long way, but now that Ken brings up directly into the substrate, especially as you're trying to get plants established and rooted, it kind of makes sense to look more into that. So yeah. It's a culture your own kit. Ken has a bunch of the canisters behind him filled up. It's just a little smaller size.
I like Crypt Keeper's question. Is the starter needed, or can you just put the bacteria naturally appear over time?
Good question, because they are ubiquitous in nature, these bacteria. The problem is they don't seem to compete well in a captive environment. Partly because our aquarium systems or ponds or aquaculture systems, or definitely like aquaponic systems for that matter, are unusually eutrophic. So a lot of nutrient and organic carbon availability. So that favors other bacteria. These bacteria can survive in a captive system just fine. The problem is they get outcompeted by other groups like Alteromonas and stuff. So you lose a lot of the benefits and natural associations between other bacteria when these guys are gone. So yeah, that's kind of why we developed it. A lot of people, I was talking about that with somebody today. They were like, well, so these don't always survive in an aquarium? And I said, absolutely not. And that is exactly why we add them.
Yeah.
Because they're naturally abundant in healthy natural habitats, but in an artificial habitat like an aquarium, they tend to get outcompeted over time. So we think of it as like a reset button. You add these bacteria, and then those guilds, like I described earlier, are more likely to emerge and stabilize and persist. So,
so Ken, I've actually realized that on a lot of because I always look for your bacteria on a lot of those aquabiomics tests on the reef side, because it's a test that you can send in a water sample and it'll kind of tell you the DNA classifications of what's in there. It's a kind of a new science technology, but on the reef side, he'll do a scan once a month on it. And you can always tell, I can always tell based on my customers, because I know that a lot of their tanks, which ones dose your bacteria and which ones just kind of go with the natural balance of the system. And yours is always surprisingly low in the natural systems.
Low in bond.
In the
yeah. So aquabiomics, so our bacteria actually don't show up as Rhodosudomonas. I haven't sent samples of the other ones yet. I'm going to, but for whatever reason, if anyone on here uses that service, they actually show up as Bosea genospecies. So it's like an umbrella group that's related to these guys, but they don't even belong to it. It's almost like kind of a slight misidentification. But at least if you know what you're looking for, you see Bosea genospecies on there, then you know that they're present. But I should actually show some of the, if I can. You guys want to see around the?
Of course. Yeah. While you do that.
Do you have to have the starter culture, or can you just dose the bacteria straight into a syringe into the tank?
Yeah, you could. So this is ProBio. You could just syringe it right out of the bottle. PNS ProBio, and kind of that orange color, right? And then at the bottom, it's more red, the carotenoids. You can kind of see where they settle. It's like blood red, but just shake it up, whatever. The young cells are motile, the daughter cells, as they call them, and the mother cells they lose the flagellum and they just sink. So they form like this sludge at the bottom. So if you just shake it up.
How long can they survive in that bottle for? Or how long can they be stored?
The longest we've tested is 1.5 years. But this right here is a culture. These bacteria actually isolated out of Boulder Creek, and they were bottled for 3 years. And I just inoculated this cube, and I'm waiting to see how it turns out. So far I can't. Now even looking at it, I think it's growing, but I'm waiting to see how it turns out. But this would be 3 years in a bottle, but at least 1.5 years on the products we sell.
Do they have to be kept under light?
Yeah, like you said, they're tough. They're hard. They're resistant. What's that?
It looks like you keep them under light. Do they have to be kept under light?
They don't have to, but grow roughly twice as fast when they can. The light. Yeah, they can grow roughly twice as fast to synthesize. So, but you can keep them in the dark indefinitely; they'll survive. The one that we kept for 1.5 years. Actually, it was a customer. They just lost a bottle, and then he asked me if it was still good, and I'm like, I don't know. Send it to me. I traded him 2 new bottles for it and used it to test. and grew a new culture with it, and it just like took off like wildfire as if I just made it. So my guess
Awesome.
in the literature, you can store them at room temperature for like 3 years, these cultures. So
Nice.
so we got all three. So this is Rhodopseudomonas. This is in really going to be hard since I can't flip it. The one that's in
Wi-Fi seems to be cutting out a little bit too.
Oh, sorry. It's probably just because I'm moving around, but this is the Rhodopseudomonas, and then Rhodospirillum. Are you still hearing me all right?
You're cutting in and out a bit. It's getting choppy.
Okay. How's this
It's all
again? We good again?
Almost. You're frozen. You'll come back in a second.
Okay. Maybe I'll just bring them over here, but if I have apples, but the other one is Rhodopsirillum, the skin one we do.
On StreamYard chance.
What did you say?
It looks better online.
That's
than it
mixed
does on
with
StreamYard.
Okay.
Oh, never
All right,
mind.
cool.
It's just as bad now.
Okay.
Rhodopsirillum is mixed in this other product that's kind of purple, but I guess it doesn't really show up in cameras sometimes. It's like the magenta color that you see on the front here, or even more pinkish, but the colors don't come through on the camera. But that's Rhodopsirillum. That one is actually, you were talking about like using these in like interstellar like vessels. There's actually am I cutting out?
You cut out, and I'm really curious.
I know it's a great way to cut out. I think he's
I
just.
know this is like the most suspenseful like part of the whole episode right now. I'm so curious about space bacteria.
And I think he might just be sitting there still and goes.
I know he's just playing with my emotions.
I've actually been doing a lot.
Right. So it's called the Melissa Project.
Shrimp in space.
Oh, there we go.
Can you all hear me?
Yeah. Kinda. Now you're gone again.
Can you all hear me now?
Yeah. Now we can. A little bit. And now we can't. It's okay.
Yeah,
Figure
great.
it out.
I think he can hear you. Tell us about the shrimp,
Yeah.
or tell him about your space project.
Yeah, talk about space shrimp.
Well, we were
shrimp.
trying to send shrimp in a rocket, but the whole mission was a failure. I guess they had engine issues, and then the next day they had weather issues. Then the next day after that, they couldn't get clearance because of insurance issues. So they never were able to send the shrimp up in the rocket. And so I'm going to do a video on it because hopefully it's not the end of the story. Maybe they'll try again next year.
I still say we launched the shrimp.
Yeah, maybe.
I know, right?
We'll see. Maybe I'll make enough money from Triple Crown and be like, yeah, let's get a rocket. But probably not. Money from Triple Crown. Yeah, let's get a rocket. But probably not. Money from Triple Crown. Yeah, let's get a rocket. But
Whoop. We got some-
I'm gonna mute him.
I saw
Probably
something
an
about
echo.
space shrimps, and I think that's where we.
Yeah.
Whoa.
Why is that working?
Trying to figure this out. We've got like
We got our IT team.
technical difficulties here. We're picking up the echo hardcore. Let's try this.
You might be back though.
I know I'm trying to unmute him now. There we go. Oh,
Yeah.
we're getting an echo from your from something. I don't know what's going on. We've got like
We got our IT team.
oh wow,
Difficult. Take a look at the help section
Okay.
in your Alexa app.
wow. There's a hardcore echo there. Who's using Alexa?
You said the word and it went off.
I triggered your Alexa.
Yeah. But that's not its name.
Oh, what's its name?
Oh, what were just happening? What happened with Ken? A reverb.
Echo. Oh, I get it now. That's
You're having
funny.
trouble connecting to the internet. Take a look at the help section in your Alexa app.
I forgot that was what his name was.
That's too funny. I figured it would have been strained to like your voice, not my voice. You're creepy.
They definitely
Oh,
know
they said, Echo, play Rocket Man.
it. You're gonna make it happen.
Hey Echo, play Rocket Man.
All right. Well, since Ken's not here, Chance, are we doing Aquashella Dallas scaping contest? Yay or nay? I need to know.
I mean, I thought you were doing it, right?
Do you want to do it too? I need to know to put you on or not. I haven't made up my mind yet.
I haven't made up my mind yet.
Well, it needs.
I've made up my mind.
I know Brian's not going.
Give me till the end of the episode to make up my mind.
Okay.
Ken's back.
Get it?
Are you there?
That's funny.
Oh man, we're still echoing. Ken,
Yeah.
I don't know what's going on, man. We're having like really bad echo
It
and
was.
everything coming from the screen.
It's funny. I figured it would have been
we-
straight to like your voice, not my voice.
Extremely appreciate you coming on.
Echo,
if
play
you
Rocket
can,
Man.
Send us some. There we are. Hey, he's backstage. Here we go.
Hey Echo, play Rocket Man.
All right. So, gosh,
Ask
I
him
can't
to
even
watch the
think.
windows
All right,
open.
well, since
All right.
Man.
So,
He's trying.
if you can hear us, Ken, we're sorry. Something's going on. I don't know what it is, but we really appreciate you coming on. If you would like,
That's
please
wonderful.
drop a link to your website in the chat. If you, that would be amazing. That would help a lot of people out.
You got to put it in private chat, and then Chance can copy and paste it into comments.
Yeah,
Can't
yeah,
post
that's.
links because he's not a moderator.
Oh, I see. All right, yeah. So put it in private chat, and then I will
Yeah,
copy and paste it into the chat.
I had questions about snails and activation time, and
Oh, I'm sorry.
I was just getting into
Hold
it.
on. What do you?
Well, we can. What's your? I don't know. I probably wouldn't be able to answer it at all.
I was going to say, throw it out there and see if we know.
Maybe he'll chat it to you. Maybe
I can
someone
call
will
Ken
know.
on the cell phone. I'll just hold it up like this.
I'm trying to find private chat to even figure this out. I
If
feel
you
like
look on the right hand side, there's comments, private chat, and then there's also like the little man, it's like a paint board thing for all your options.
Oh, I see it now. There we go. Nope. Okay. It was just you telling me how to do the mod thing. All good. Whoa. All right, so guys, what have you been up to this week? Let's get back to that. Let's try to recover from the technical difficulty.
Haven't been doing much. Catching up on water changes.
The 100
There's been
tanks.
so much rain, I can't go and buy the wood to build the next tank stand.
Oh yeah, because it's gonna just get all wet.
And then not only that, but like I need like a nice dry day to cut it all so I can bring it inside. I was also going to paint it after I cut it all, so that way we didn't have to paint it inside. It'd be super nice and easy. And then I don't know. I'm still debating on whether I want a 2x4 in the middle of the 10s kissing or put 4x4s. I'm definitely going to use 4x4s in this build.
Yeah, 4x4s seem like they might be double the strength of a 2x4. Something about math or something like that.
Adds
I just
up.
don't know if it's needed. I've been debating it all day.
I used to know. So my I used to know but I don't know how. Well, I do know how, but it's irrelevant. The actual downward pressure a 2x4 could take before snapping, and I think it's like
700 pounds.
It's ridiculous. It's like
Correct.
1,000
absolute.
pounds.
It's ridiculous. It's like 1,500 or something like. It's absolutely ridiculous.
Each one's rated for 1,000 now. Maybe back in the old days it was 1,500, but I just built the freaking stand for 110-gallon tanks, and that was one of the big concerns. Was it weighed 10,000 pounds, and I was like, I got 10 supports. It's good.
We're there.
And then Shelby's got Cory cat babies.
Nice. Congratulations. I'm gonna try to add Kin back in really quickly. Is that okay?
Yeah,
Let's
no.
try this. No.
Yeah, 4x4s
No.
seem like they might be
Oh,
double the
wait.
Didn't work.
Didn't work.
something about.
All right. I'm sorry, Ken.
I'm not sure if he's like watching
said-
on his phone or something too, and that's what we're hearing the echo.
Maybe. I
sent
don't
the
know.
link in the
Yeah.
private chat.
I don't know if maybe he has two things open. I'm
'Cause
not
I saw
sure.
he commented, and he commented, and it was a.
Yeah, he's on the YouTube. It looks like as well. But
So maybe it's his phone on, and that's
maybe.
what's lagging behind, causing issues.
Maybe. Omid wants us to build a rocket out of 2x4s. I don't think that's going to work. I mean, Elon Musk is building stuff out of like stainless steel that weighs like millions of pounds, but
I don't know if Grant
Maybe.
attaches one of those Fluval CO2 canisters to it.
Oh,
I got a big old 50-pounder over here.
I got a scuba tank. Just sledgehammer the end of it and let that be.
What could go wrong with 2x4?
We'll have to go to a park. We could use like JB Weld some fins on it, and just we'll put a
I'm
bag of
down.
shrimp on the end of the tank and just
Lick our
Hit the-
fingers, put it towards the wind.
we'll
I'm
Joe
down to
Dirt
like
the
get
top
a
of it.
hyper rail built and just launch it magnetically.
I'm just thinking we should get one of those Hobby Lobby rockets, like the model rocket things, and put
We
like
need one that at least goes like 10,000 feet.
yeah, we'll get they make like the giant dual boosters and stuff like that. My pops and I used to build them. They used to go like
it also
mad
has
high.
to go through a certain amount of G forces. So I think the rocket was
We'll
possible. It
we'll
could
duct
have
tape a
gone-
bunch of rockets together.
Yeah, I have a hobby store down the road. We'll have to go check it out when we get back from Aquashella. I got no time till then. I'm like crunching, trying to get all the water changes done in the house before we leave, and then the pond's giving me issues outside, but just getting ready.
Pond's giving you issues because of the damn photocycle of the sun is mad crazy right now. I'm getting
No,
issues.
Yeah.
It's the overflow. My filtration barrel is getting clogged, so I got to clean it out and put in new filter media.
The sun's cooking my stuff right now, man. I gotta this year. Once the shade cloth is ripped, I'm changing from 40% to 70% for everything because it's just summer's brutal, dude. And I don't have any trees or like natural shade, so my houses are out in the sun like all day.
We've been getting a decent amount of like rainy days, so my plants have been taking off and loving it. And I also had that big shade cloth. I don't know what percentage shade it is, but it's pretty nice.
Yeah, no, yours it's thick. It's like a sun sail. It's like one of
Yeah.
those big old sun sail things. You walk under and it turns you into a Smurf.
And we went
Let's-
to Rural King and we got a muck bucket. Put some
Nice. Rural King's cool.
jungle val in it.
Dude, Royal King has everything. Like almost too much stuff.
I'm probably gonna get a couple 100-gallon stock tanks and throw a bunch of floaters and other plants out there.
Do it.
Make some frog lagoons so they breed in there instead of my shrimp tanks. But I'm probably still gonna put shrimp in them. So
Just
Hey,
just put some like carnivorous cichlid or whatever that doesn't eat plants in there to take care of the tadpoles, and he'll tend your shrimp. I get a bunch of leaf fish. That'd be cool. Just put a bunch of African leaf fish in there and let them wipe out whatever's in there.
Hey,
Yeah.
Chance, can you put up Jamie's comment?
Which
The one
one?
that-
Oh, about the balloon?
Yeah, that's the solution right there.
I think you got to get like FAA clearance to do shit like that, though.
Not in Florida.
Just don't write
This
your
is
name
Florida.
on it.
This
We
is
just
Florida.
do it and then ask for forgiveness.
Yeah, in a federal pound me in the ass prison. No, thank you.
I know a couple of guys that got busted by the feds on importing corals, and that's not a fun type of thing to go through.
Yeah, dealing with the law in any instance isn't fun.
Yeah.
So,
They just passed a law now that you have to report any injuries and escapees for class 3 animals in Florida. So
Yeah.
like a lot of people like get bit by their poisonous snakes and then don't report it and stuff like that, you know?
Yeah.
So, oh, I didn't get bit by my diamondback rattlesnake. I got bit hunting for him out here, you know? And it's like, come on, man. You got 15 of them back there. You know you got bit by one of them.
Well, you know
Yeah.
they all know who to go to for the venoms, the antibodies.
yeah, a lot
You
of
know
them
how much
have,
it costs?
well, a lot of them have people who
Depends on
have
if you go to
the
the hospital
antivenom.
or your buddy.
Like I know somebody who lives close to me who knows people who has the antivenom and doesn't have to go anywhere, but luckily he's never been tagged, so it's like
Yeah.
he doesn't have to worry about that
Do you
anyway.
know how much it costs though?
Probably a lot, man. It's
They're
hard
like
to cultivate.
60 grand. 60
Yeah.
grand. You know how they cultivate it?
They milk a snake and they spin it off and stuff and let it eat flesh. No? What do they do?
They get the rattlesnakes to, or whatever type of snake, to go after horses or cows, and then they build up the antibodies in their blood, and then they isolate it from the cow, sheep, other animals that they use.
That's nuts.
Yep. That's why it costs so much money. And then it also doesn't last long. Like it's got a quick expiration date.
So I know you can also do that to yourself. Like you can desensitize your body to certain venoms of snakes.
Yeah.
And basically these dudes, they inject themselves with the snake's venom in small amounts over like years, and they can actually become immune to the venom that's in the snake. But apparently like it leaves giant cysts and welts and like shit in your arm or wherever you get the injection at. Like it's gnarly. You got to really be committed for that. Can you imagine if you had to do that to breed
Not the
shrimp?
Princess Bride.
You got to like if shrimp are poisonous, in order to handle them, you got to like inject yourself with shrimp stuff. I don't know, man.
Ken's backstage again. On different
Yes. Let's see this. All right. Hi, Ken. We're going to try to throw you on here really quickly. Hey, buddy.
Hey.
Hey, it's working.
Good now.
Rachel.
All
All
right.
right.
It's like night and day. Way better now.
Way
nice.
better. The image is better, too.
Good.
Much better.
Sorry about that.
That's okay. That's okay.
Oh, no worries.
Yeah,
We're
no
not
worries
very
at all.
formal here.
Yeah, I left off in a weird spot too, so I just had to jump back on. That was
Space bacteria. We were waiting for it.
Yes,
It's on the
yes.
cliffhanger.
Space bacteria.
No, space shrimp.
space
he's
shrimp,
space
right?
bacteria.
No, they have there's a project called Melissa where they're using rhodospirillum, the last bacteria I was showing you.
Is
They're
that freshwater or salt?
both actually
Okay.
can survive in either, and they are using them for nutrient recovery on or experimenting with these systems that where you recycle nutrients, kind of like you were saying, Chance. Just I don't know if they maybe even as food, but just to like recover wastes and stuff on vessels for manned missions to Mars. So they almost, because of the length of time it takes to get to Mars, that people will almost certainly have to grow their own food on board, and that's like one of the things they want to implement in their like little I don't know how it would be like hydroponics, like in space or whatever. You got water like all over the place or what, but contained, right?
That'd be awesome. I don't know. In my
So-
head, I just see like droplets of water with plants growing out of the droplets floating around everywhere. But
That's
it's
yeah,
contained in
similar
like
to
a
Grant-
a
That's
flat
similar to
bag.
Grant's project, the space shrimp.
You know what the big issue though is with Mars and hydroponics is that because of the lack of atmosphere, the boiling point is not like the point at which water boils. It's a way lower temperature than it is here on Earth, and then the point at which it freezes is higher than it is here on Earth. So there's a very, very small temperature zone that the water can be used to grow hydroponics.
So have you ever heard of film farming?
Thin film.
Film,
Is that where they
yeah.
use
Yeah.
the fog?
It's film. No, it's just like a film, and it requires like next to no water or nutrients or anything.
Yeah,
I wonder, like,
that's why I thought it was fog. They literally just put a fog through. They don't use water.
I don't know. I just know when I've seen it done, it's like just like there's like nothing. It's just a little moist film that the plants just continuously grow out of. There's like no additives to it at all. But it could be because of the fog or humid environment of wherever it's at. But I wonder, like, the applications of that with the bacteria that you're speaking, you know, and stuff as far as symbiotic non-water systems.
Yeah, I don't know much about the system. I'm sure it'll be pretty weird if it's on a spacecraft and
Right.
if it's on some kind of like pressurized module that they build on the surface when they get there or whatever to mitigate like what Grant was saying, but you know, it'd have to be under pressure or whatever to even work. But
They need an electrostatic surface to hold the water to it.
they need aquachar.
Yeah.
Yeah.
Uh,
Actually, shrimp. While I have a chance to talk to Grant about this, these bacteria are actually used in the shrimp industry pretty extensively. They
Aquaculture.
it's like shrimp, like for food shrimp, but I mean basically like a lot of crustaceans suffer from the same types of disease, and there was a Vibrio that was wiping out the Pacific white shrimp industry, like just decimating. There's some kind of hepatic, like a liver or kidney or something like that. That I don't know shrimp biology that much, but I can't remember what hepatic is. Is it liver? Or no, it'd be kidney, whatever. Yeah, it's
hepatic would be kidney, I believe.
Kidney. Yeah. So apparently shrimp have like an equivalent of a kidney, and these this particular type of Vibrio would attack it and just wipe out shrimp like crazy. And they've discovered that these bacteria are so effective in warding off Vibrio that now it's just like a routine probiotic that they use for the Pacific white shrimp industry. But just the fact that they form biofilms, and I know like a lot of the shrimp you guys raise would feed on the biofilms, and it would enrich them. The carotenoids would help color, at least some of the reds and oranges and yellows. It'd bring out the colors in some of the shrimp.
It's liver. I was wrong. Sorry, liver.
Liver.
Omid's a medical professional. He would definitely be the one to know. Liver.
Yeah. So I guess shrimp have some kind of liver, I guess. And it's prone to infections. So I'm imagining that'd be all types of shrimp. So it was Caridina or
the carotenoids interest me too, because like in shell development and stuff like that, like I wonder, like you were just saying, pigment and shell development, if you could like really amp up color, like Grant, that'd be a cool little experiment to take like low grade culls of X color and just like boost the hell out of the water with this bacteria and see what happens down the line genetically.
Yeah.
Not even genetically, but just pigment wise rather.
Mm-hmm.
Yeah.
That would be cool. He missed the question. He totally missed it.
I missed it. I'm so sorry. I was distracted with other questions. Oh, my bad.
It's all good. So
no, we were talking about using it as a probiotic for your
I was sold on it. I figured afterwards I was gonna get my hands on it one way or another.
Yeah, I think it'd be cool for you to just try it out with like some really low grade culls of a certain particular color,
And see what comes up.
And see if it improves down the line, or even as age progresses in the shrimp, like just to see if that plays any kind of role in shrimp pigment. That would be awesome.
Yeah, no, I'm for it.
Do you know of any cases where people use like astaxanthin or something like that, where they'll add it to a shrimp diet to?
Yeah, it's in a couple of like different colorful shrimp foods where they're trying to like increase the red color and in the red cherries and stuff like that. But bacteria themselves is something like all of yours are going to be liquid bacteria, no powdered at all.
Yeah, they're like complete. They're actually living, so they would just
Yeah.
be embedded in like the biofilms and stuff that there are papers where people have made them into feed or put them into feeds.
Yeah.
And they're beneficial that way too. They're still nutritional. They're like in some cases over 70% protein, and have a lot of B vitamins, some D and E. But it's the carotenoids that really make them valuable, because astaxanthin in particular is just really expensive.
Well, dried bacteria powders are a big thing where people will usually feed those for like a source of food for baby shrimp, mainly because like they grow so much of the biofilm, as they're being dosed.
Cool. Is it like a dried? Is it just like in a spore form where they come alive, or do they just? It's just like a powdered food.
The most popular product is Bacter AE, and it's just like a white powder, and you put it into the tank, and it's like, I think a known thing in the hobby. It takes at least a week for the bacteria to come out of the dormant state and start cultivating.
Okay.
So I didn't know if you had something like that, or if you were able to cultivate something like that.
No, that's a spore. That might be. I'd bet anything it's bacillus. That's a spore former. Someone could look it up, I guess. If
It's bacillus. I got tablets
of the same of what Grant was talking about.
It's this is it is the same thing Grant's talking about, but this is with my bacteriologist from Holland.
Okay.
He's got a slow release tablet that slow releases the bacteria over time as a.
What were to happen to your culture if you were to like dehydrate it?
They die.
Yeah, I'm just saying, like, you can't bring them back to life by hydrating them or anything like that.
No, not these guys. There's one there. It's but I haven't even been able to find where I can obtain it. There's one Rhodospirillum. Well, is it Rhodospirillum centurium or something like that? It's like one purple non-sulfur bacteria I know of that forms like an endospore, and it can survive desiccation, but it's really rare in this group.
But because it's so enriched with vitamin B and protein, it wouldn't be like a good thing to dry out and maybe farm for a food source.
Oh, it would, and there have been studies showing that it would. They definitely want. There are a lot of people that want to do that. It's just that they can't be regenerated where they would come alive again.
What are you feeding the bacteria?
Depends on the product. So there's ProBio. That's actually like where we were talking about the water milfoil, where we make the extract. They consume that. But then there are other formulations that's our herbal formulation. And we have what we call our herbal formula, or our mineral formulations. And those are just like a pile of different salts, like, ammonium carbonate and all these different vitamins and trace elements and salts that basically it's almost like a hydroponic mix or something, right? Just a bunch of nutrients.
Okay.
And that one, in the end, the bacteria consume all that, and then what you have is mostly bacteria left. So, like for the difference between that and if it's alive as opposed to like a powder, or certainly like if you're just buying like astaxanthin and mixing it into a food, there's something about carotenoids, and I honestly don't know a lot about this, but from what I understand, carotenoids are only lipid soluble. So when you feed them to something, it's better if it's like a whole cell diet.
So like THC.
What's What now?
So like lipid soluble, fat soluble. I was just.
Oh right. Oh right. Yeah. Or like a lot of the vitamins we take or whatever. But it
Right.
you have to. It's better if it's taken in with the other foods, like the fats that will like deliver it into the digestive system of the, I don't know, whatever, how that works. But I just know that the carotenoids are more bioavailable to whatever eat them if it's like a whole cell diet. So for like shrimp or something, I don't know if the powders would work necessarily, but it would definitely work if they're eating it directly from a living biofilm. So
Yeah.
So what if soaking food in your culture, like culturing, like or making even like a gelatinous form of this food to where it still holds moisture but not saturated moisture? Would it be able to live in an environment that is gelatinous?
Probably, but most of those gels you have to heat them and then cool them. I think the heating process would kill. If you found a
What
gel
about post inoculation or post heating? So like
yeah, maybe I suppose if you mix it in, but you can just soak a pellet like a
okay,
like
that
a
would work
food
too.
pellet. Like I think there are probably a lot of shrimp foods like that, right? Like little pellets.
So you'd have to sell it as a 2-part thing, essentially. Then.
So here's the thing: is the way to use Ken's bacteria as a food. So you got to think of it like a live probiotic. So you're getting a live cultured bacteria. It doesn't take much. So if you were to take like your food cup and put a little bit in there, it not only will it get on the food and find a home on the food, and that's what your shrimp will eat. It'll also, whatever's not eaten, instead of rotting in your system, it's either going to end up in your substrate or in your filtration or anything like that. So it's not going to waste. So if you add it with the food daily, whatever's not consumed still will prosper in your system. It'll find its space in your system.
Good point.
That makes good sense. That's a good way to think of it then.
In the shrimp farms, that's actually what they do. They considered it both a water remediator. So it was used to actually improve the water quality, but they put it in the food, and then it actually had the probiotic effect while it was in the animal gut. So it's kind of like a twofold benefit.
Yeah.
Sorry to go to chat real quick. I guess this would be you, Grant, because I don't grow the hooker moss. You are the hooker moss man.
He's our hooker man.
Have you guys looked on anything for Prime Day? Because I haven't at all.
I just got the freshwater master test kit, and somebody posted that link. I didn't even go on to look. Everything I need right now is at Home Depot or Petco.
Right. I haven't.
Gemco.
I run T5s and stuff like that. So
I got like 8 of those back in the corner.
But the Rotala, I'm not sure.
Yeah,
I mean, just go for full spectrum LED lights on eBay.
If you're looking to get color from the Rotala, then you're going to want something that's got a lot of PAR, because it's anything that you're going to drive color from is going to be photoreceptive, so or nitrate reduction. So
buy 2 of them.
Either way, a lot of PAR. But mosses aren't going to respond well in super bright environments all on their own, because then you're just going to have surface area for algae to grow on, because they grow slower than plants do. So you kind of have like 2 different goals there. But just a medium strength full spectrum light will give you decent growth on your Rotala. You won't get amazing color, but it won't fry your moss either. But I haven't seen prices of anything, so I have no clue, man. Sorry. Here you go. Somebody answered. I should have just read further down. That sounds like a good recommendation. I've been seeing the name of that light all over the place. So
yeah, I've heard
that. So with the bacteria, I guess my question, my last like curious question is the milfoil versus the mineral. So your natural versus the mineral. Have you had any studies to see the benefit of one or the other?
Not really, because I'm the only one that I know of to ever grow it on water milfoil. But they grow it on all kinds of wastes, like so it's they call it downstream waste recovery. If you're so like in olive oil pressing plants or stevia waste water, stuff like that,
Mhm.
any kind of like industrial organic waste, they could use, many cases, use these bacteria with or without little additives if they're missing a trace element or something. But I just happen to use water milfoil because obviously, the obvious advantages to the environment by removing it. But it turned out, because of that telemigrandin 2, that chemical that it produces, the plant.
Right.
So Myriophyllum can be like, it was like 15% dry weight telemigrandin 2, just that one tannin, 15% of its dry weight. That's how much of that stuff it produces. It's just insane.
So the ability to be able to isolate and use that in an aquarium would be huge, as I was saying before, to be like just a prophylactic to algae. So as you would dose the bacteria, you would be dosing this telemigrandin in the same sense. So
so I don't know. I've never separated the two out to know if there are any like secondary benefits from any undigested telemigrandin 2 that's left over in the culture medium, right? Or if the effects are just competition with the live bacteria. I haven't separated those out. I have, however, like used teas from the telemigrandin 2, and I was just curious about its effect on algae and bacteria. I had a green algae. I think it was a desmid algae.
Okay.
So it's like this green algae I had, and I took like a bunch of like mason jars of the same size, and put some and put the tea in some of them and some not, and it actually did kill the algae.
That's awesome, and that's what I think. In my personal opinion, that's what I'm most interested in with the bacteria game, is the natural approach to algicide. You know what I mean?
Now that had nothing to do with the bacteria, because I intentionally, in that case, just used the tea and I didn't inoculate it.
Right.
But if you're growing water milfoil, it's kind of like so the reason a lot of these temperate lakes are like super clear water, where they have the Myriophyllum, and other plants can produce similar allelopathic substances.
Allelopathics. Yep.
Yeah. So that's part of the reason why there's no plankton in that phytoplankton in that water, is because of these substances they release.
And that goes back to starting a plant up with a lot of plant mass, rather than putting 3 stems of one species in a 40-gallon tank and saying, all right,
and then fertilizing
and then dumping ferts in. So it's thank you, because that kind of brings things full circle. Just to talk about the allelopathy and how that's going on in your aquarium with all of the bacteria stuff. And so it's so overlooked. It's a world that nobody pays attention to. Everyone gets so tunnel vision on nutrients and CO2 and light. That's the only thing in this hobby, that questions that I get. And when I try to talk about, take care of your substrate, how is your filter looking? Things like this, they want to think that I'm just calling them like lazy aquarists, but really I'm giving them the answer to their problem. Let your bacteria have a place to actually colonize and live. Take care of the small world that feeds the big world. And, you, in your research and your basically life path, has kind of just, checked that off the box for me, to where I don't have to worry about trying to prove it to anybody anymore.
There's a lot of research actually that's been done on these bacteria. So I walked into this. I'm not a microbiologist. I'm an ecologist by training. And that's why I found these particular bacteria interesting. But it's not like I know everything about all kinds of bacteria. I've just really focused on this one area.
Yeah.
But it's just that holistic approach I think you're kind of talking about is something that we may have had like years and years ago in the hobby, but we're getting more, or people get specialized in certain areas and like almost get tunnel vision and forget that it's an actual ecosystem.
Yeah, and Ken you and I have talked about this for a long time, about the water milfoil and the carotid, like what you're doing with the teas and everything like that. And Yeah, I definitely think that it's not the easy route, and a lot of people don't realize that, sticking by a product that's not the easy route, and that you're putting your time and energy to. Now Chance has, when people ask these questions, he has a product to send them to, so that's awesome.
Exactly.
I got a question on snails.
There we go. He was. That's right. You have a snail question.
Oh yeah.
How would like some Malaysian trumpet snails do for the bacteria? Would they be beneficial, or would they be consuming and probably destroying the colonies as they're sifting through the substrate?
More likely the latter, yeah. Not only eating the bacteria, but aerating the substrate. And, they're less competitive with other bacteria with increasing oxygenation. So that turnover would actually kind of inhibit them.
Not tonight.
Or it could just drive them deeper. In that case, you'd have to have a really deep substrate then.
Those Malaysian trumpet snails go everywhere.
Yeah,
They do.
Yeah.
They go everywhere and anywhere, and once they're there,
But honestly, that's a good reason to dose his bacteria, because if you got them in your tank and you don't have that layer of substrate, or it's getting oxygenated a lot, at least you're giving it a fighting chance to have your substrate have that layer of protection.
Yeah, and no matter how much the substrate itself gets disturbed, the bacteria can potentially inoculate areas actually where they're most important too, like at the plant root. They'll live in like little pores right on the plant, like on the root, and you can turn the substrate all day or not even have a substrate, and they'll still survive just deep in those little those anaerobic pockets, right up in the site where they're most important. Getting those leachates from the plant root, and they can just deliver that fixed nitrogen right to the plant directly like that. So,
primarily how epiphytics drive and such, in a way.
Mm-hmm. Yeah, it's all pretty localized. So it's just a matter of growing. It's like most people use these at least on the saltwater side. What they're really trying to do is it seems like everyone's always trying to get to lower their nitrate and phosphate levels.
Yeah.
And you need a lot of these bacteria to do that. So it's not like if you're just trying to grow them on a plant where they're very localized and that you don't have a lot of them, have to have a lot of them. And they're also very competitive because they're in their element on a plant root, whereas in the open water in a reef tank, they're just not. So, honestly, these are really a freshwater. Most of them are freshwater bacteria anyway. The rotobacter I was talking about is strictly freshwater.
Now the other thing in the shrimp hobby is like there's several companies that'll say, "Oh, you don't want to dose several different types of bacteria because they'll compete with one another." But it seems like you want a couple different types of bacteria. Yeah.
Guilds, like I was saying, they actually serve each other. Like very similar bacteria, if they're in the same niche, then yeah, there may be some competitive exclusion there, but so what? It's like then you just hedged your bets with two different types of bacteria to do the same thing, right? If one didn't survive, maybe the other did, but it's still not a bad thing. You spent a little more, but you hedged your bets, and at least one of the two survived. That's better than nothing, not even filling that niche with anything.
Because some of them have like 7 different strains of bacteria in one, bottle. So it's like
they're usually inactive though.
Yeah, well, so you'll see like consortia like that, and most of those are meant to act together. Like Bacillus is another widely used probiotic, especially in the shrimp industry. And I think it was shrimp in the study I saw. The like Bacillus and Rhodopseudomonas, the purple non-sulfur they were looking at, actually worked synergistically inside the animal's gut. Like both species of bacteria act as probiotics, but when you put them together, they actually have an even better effect because they benefit each other as well as the host, if that makes sense.
And do you have both of those available, or do you just deal with the purple non-sulfur?
So far, exclusively purple non-sulfurs, but I can
get you some Bacillus.
Yeah, everybody like uses bacillus. It's like
That's your most common bacteria that you're going to dose in your system is going to be the bacillus strains because they can transport and have a long life.
a lot of fish foods even have it in it already. So
Yeah.
yeah. Bacillus and lactobacillus are. If you see a fish food that says contains probiotics, it's almost certainly Bacillus subtilis. But yeah, that's a good one to have, and it wouldn't interact with these bacteria in an adverse way at all. In fact, they'd actually enhance each other's activity and increase the benefit. At least in that one particular application, that's what that study proved. But you see those together in plant. So, Chance, you may have heard of EM1. It's like a mix of, so it's a formulation for
hydroponic, or
yeah.
I haven't dabbled too deeply into hydroponics, so
Okay, that one is it was developed by a Japanese gentleman. I think it's actually the EM is like a protected name or whatever, a copywriter, but basically that method uses purple non-sulfur bacteria, either bacillus and or lactobacillus, and then yeast, cerevisiae.
food source.
Yeah, but the three act together. You know, they all do. They have this like synergistic effect on plants.
Yeah.
So, which is kind of funny. Like plants or even animals, you'll see those same bacteria, all that same grouping used together for like fish and shrimp probiotics. So it's a really generalistic kind of association that they have.
Right, and it really and it should have been obvious all along for us to be adding these to our aquariums with maintenance. You know what I mean?
Yeah.
It's something, I don't know, like I said, overlooked, and it's nice that there's the light being shined on it a little bit. So
we've known about it for like about 40 to 50 years. We've been studying these pretty intensely, actually, mostly in East Asia, and they've been using them in aquaculture in their shrimp and fish farms for decades now. So it's almost like the West is kind of behind.
Oh yeah, well, and the aquarium hobby in general, it takes a while for science of horticultural science to cross over into the planet tank world.
Yeah.
People try to, battle methodology with each other. It seems to become like a battle of egos rather than like, look, here's a paper saying it worked over here. Let's try it. You know what I mean? Yeah. And it's nice, again, this bacteria thing I think is going to be game changing for the planet tank world, especially if the right people start picking it up and start talking about the actual importance of stability in the bacterial world in their tank. I think it's like the next evolution in the hobby as far as what everyone should start focusing on, rather than this nutrient tunnel vision or fertilizer tunnel vision that everybody has been in for the last 15 years. So
I think especially if people start using services like Brian was talking about earlier, the aquabiomics, where they actually analyze your microbiome and tell you, they give you a report, and they basically tell you, like, might be at the level of family or genus, sometimes to the level of species, what all the different groups or species of bacteria that are in your tank. And I think maybe the freshwater people, if they catch on to that and start using services like that, they'll see differences. It doesn't mean anything unless you're into bacteria, right? Specifically, until you start to associate differences between different people's tanks microbiomes with different states of health. So if someone's having like kick-ass like success with a type of shrimp, or with with their plants or something like that, and their microbiome looks this way, and then someone else who's having kick-ass success, their microbiome looks the same way as the other person, but the person who sucks and everything dies and their tank or their systems are failing and their microbiome looks different, then we can start to correlate and figure out what works and what doesn't in terms of the microbiome. And then at that point, people like me would be able to start cultivating some of the more important groups of bacteria, some maybe that are lacking in captive systems, to kind of serve the hobby.
So that's a good idea. So there's some people on that I know, like Omid, for example, is a really good aquatic horticulturalist, and he's in the chat. But I got a couple other really good growers who I'd be interested in giving them each a finicky plant that I know they all can grow really well from what I've seen in the past, and then taking all their microbiome analysis and putting them together, and then also taking readings from other people that I get questions of about these finicky plants that don't look so hot, and seeing and basically proving the point as what you're just saying. That'd be very easy to do.
So one of the cool things that they found out with the coral side is some of the vendors and stuff like that have a lot of corals in and out of their tanks. I think they had one guy down here in Florida that had like 810 different strains or DNA profiles of bacteria because you add so much in there. So like, I'd be curious, with those tanks, if you were to give the stems and then get trimmings back from them and put them in a tank together, if it would increase the diversity or, one of those types of experience. Because a lot of it, nature will find a way in a lot of states, like in instances. So, but yeah, I'll let Ken throw in his opinion on that.
I would think like some of the tissue culture stuff would be really prohibitive for, in terms of using plant fragments as vectors.
Yeah.
Because just
sterile.
Intentionally so, right? Like you're kind of
has to be.
It's a shock. Yeah.
So just kind of by nature of the process, it would eliminate a lot of bacteria. Some would make it, of course, because it's almost impossible not to. But it just so many groups of bacteria, particularly the anaerobes, would be just destroyed in that particular method of cultivation. So I think the best way would be to like transfer, like substrate transfer would probably be the best.
That'd be,
Or biomedia transfer. So somebody, if they've got like a big bucket of aquachar or something,
way to do it.
That
pre-inoculated,
and then just move it around that way, and but or buy my stuff.
Exactly, that's what I was just about to say. Is or you could skip all that and feed it the right stuff that is right all the way, and just buy the product that Ken has. We put the link up in the chat there. Hydrospace.store. So if you guys are interested in it, definitely check it out. I can 100% speak to it, not only in the aquatic world, but in the terrestrial world as well. And if you've gotten plants from me in the last 6 months, they've been sprayed with this. So all the people talking about the success they've had with all the immersed clippings and how easy they're converting over to submerged and stuff like that. This may be why, or it may just be that they convert easy. But I can say that this is one of the products I use and will continue to use without a doubt, and we'll probably dive deeper into using it into more applications here very, very, very soon. So,
yeah, I can't use it on a tank.
Yeah, no, the using the what Dark River said about injecting it down into the substrate area is
by roots, yeah.
Yeah, so that's going to be interesting.
Like like with the people doing soil, I guess it's the equivalent of the soil drench, right?
Yeah.
Where you just pour it right onto the roots.
It's hard to imagine what to do that with an aquatic plant system or aquatic system. So that's why the idea is so fascinating to. I'm sure Chance, but myself as well.
I've seen little rigs where you can get like the big syringe with the fat.
I've already got it. That's why I'm smiling. I've got like this giant syringe from other works I was doing on some other things, and I even have like the long, skinny like garbage feeding tube. I guess they use it to feed babies, essentially like infants. So it's like super small, almost pick line size tube that's kind of firm, definitely firm enough to poke in between aqua soil.
Oh, there you go.
So
Yeah.
we'll see. I could just snake it all along the tank.
One thing we're working on right now is possibly a grant with National Science Foundation, and it's for we're going to look at different applications, including like foliar and everything. But the gist of it is these one thing we didn't even touch on that makes these bacteria really useful for plants. And one of the things that it's kind of why there a lot of interest in like more in high value crops like cannabis is that they produce phytohormones. So it'd be like auxins, and in this case it's indole acetic acid, IAA, and then there's another one, a levulinic acid, ALA, 3-amino levulinic acid, that promotes mineral uptake, and I think it's actually a precursor to chlorophyll, even in B12.
And these are all things that are naturally made by the plants on their own that are now being added by this. So it's just boosting their natural process essentially.
Well, it's almost like, so of course the bacteria evolved before the plants. These like land plants are actually the descendants of these bacteria, believe it or not. These bacteria are just older by literally billions of years. But the bacteria don't necessarily, it wasn't in their evolutionary history to produce a lot of these chemicals. They learned how to do it. They somehow evolved to synthesize the same chemical or some kind of similar chemical because of their association with plants. So it's really cool that, you know, they'll produce some of these same substances just to kind of pay their rent on the roots, so to speak, right?
Right.
So because the plants too evolved ways to produce substances that serve no other purpose than feeding the bacteria. It's actually a big waste of energy to produce those exudates that they release through the roots. It's believed that it's primarily for the purpose of attracting these bacteria. So
you've got to have something to make your food, you know. It's
right.
It's evolution's amazing, you know. Maybe one day we'll have that.
So
right.
It's funny that, so Ken was mentioning earlier, you know, with the wood-based carbons and stuff like that, and his bacteria having a propensity for it. And I've always explained to people with aquachar as a biomedia that it's more about that's what after a forest fire, what brings back the growth. The first aspect is the bacteria is going to take it and start breaking down whatever was left behind. So with an evolution standpoint, you know, the bacteria and stuff like that, sometimes the plants evolved around the bacteria's, what they're able to produce and what chemicals they're able to do, because going back to the beginning, everything's really lazy and they just want to eat and reproduce. And whatever the quickest way or however easy it is, the better success you'll have.
The burnt soil thing brings to mind that these bacteria are like, they're kind of like pioneer. In ecology, we call it that you have your pioneer species that are good invaders, and there are other species that are good competitors. But the good competitors show up later. They're not particularly good at invading. These bacteria are actually really good invaders. So if you have a carbon-rich environment like that, but one that's not very rich in nutrients, particularly nitrogen, right? So these guys can actually kind of kickstart an ecosystem because of their ability to fix nitrogen. So they essentially fertilize the soil, but then there's also the bioremediation aspect. If you're in horticulture, if you're fertilizing all the time, you eventually damage the soil because there's no way, you can never like get the balance just right, and eventually you have an abundance of things. They reach a point of excess where they can actually harm the plant, and these bacteria are used to kind of bring all that back into balance. So they're actually used to repair damaged soils. Even arsenic, stuff like that, they can sequester and remove from an environment. So yeah, from that aspect, going all the way back to what you were saying, Brian, about like the burnt forest or whatever. It's pretty incredible, like, how there's this like the biological succession, as they describe it in ecology, where everything kind of fills in the blank slate in an order, right? What's up, Grant?
Nothing, Brian.
That's what I ask. That's my answer to my kids when we're driving someplace.
Well, we have had your time for so long. We'll make this question that I have up on screen the last question for you, because this is the same person that asked about injecting it, so I appreciate his question, obviously, and B, I don't want to take any more of your time. It's starting to get late. I feel bad. Grant's probably going to fall asleep. So
it's not your fault, man. I've been up late nights and up early mornings trying to get everything done before Triple Crown. I didn't realize, like, now until December we have at least one show every month.
Yeah. I was just looking at the airline cost to go to Chicago, and I'm like, because I just bought the tickets to go to Dallas, so it's like, gotta hustle them plants, man.
Just road trip up, drive with us.
I might.
I won't have the kids for Chicago, so
I don't know. I'll think about it. I'll let you know. I'll probably end up buying a plane ticket. I don't know. We'll see. But yes, so he's the question, I guess, that he's asking is where exactly should he inject it? For some reason, my brain's not reading the question right now. I'm definitely going to try injecting it, but I use void space below my substrate to attempt to promote anoxic zones instead of full anaerobic zones. Would that be detrimental?
Yeah, because these guys would like to live. I mean, they can live in a microaerobic zone or an anoxic zone, but if they don't have something like something to live on, that wouldn't help them so much. So it sounds like this person built like a plenum or something.
He has an interesting soil system that he uses, and it's layered. So there's areas that, like he said, are different than other areas of the tank. So he has it layered in different ways.
Okay. Well, if you have like a micro aerobic zone above, say, like the barrier over the void space, that would probably be fine. So if you were going to inject it, I'd just go all the way down to that barrier and inject it at that level.
All right.
And that should be fine. So it's a lot better than, I mean, in most cases, people just pour it right into the
exactly
tank, right? And it still finds a way. So
I mean, I'm gonna do both, to be honest. Like I'm gonna do both just to double the odds up a bit.
Before we go, Chance, how do you water it? Do you just dilute it down in your water as you water your plants, or is it a misting bottle? How do you use it when you use your terrestrials?
So I use a backpack mister that I mix the solution in. That's for foliar feeding. And when I do dose the soil, it's just a regular watering can that I've mixed down. So nothing special.
Yeah, I was just curious.
I'm setting up an IBC tote, though, actually, here soon, that will be ran off a diaphragm pump to run the misting system. And in that IBC tote, I'll have my full barrage of foliar food. I'm going to actually see, I don't believe it caused a problem with the bacteria living with the fertilizers. So we'll see. Do you think that'd probably be okay, right, Ken?
What's that? I'm sorry.
It's
I
all
was
right.
just posting something, actually.
No worries.
I just posted a paper for anyone that's interested. It's basically a review of the literature, I think from like 2021, which is very recent in terms of like science. It just kind of provides an overview of all the uses for these bacteria with plants. So if it doesn't stick, let me know. I know YouTube can be finicky if you're trying to post a URL. You know what I mean? Like
it didn't even show up, but if you send that URL to me in private chat, I can post it. I don't have any mods set up, so I don't have any way for anybody to post links other than me. I don't know how to make mods yet. I haven't done that.
okay. Yeah, right on. I'll do that. What were you saying?
Just
nutrients. Is it if he puts it in a container with nutrients?
Oh yeah, okay. Oh yeah, if I so I have a foliar feeding like system that I use, and if I mix that bacteria in with the nutrients that I foliar feed as well, because that's what I've been doing, I haven't seen any issues. But I'm talking long-term storage, so like a 300-gallon IBC tote or a 275-gallon IBC tote, with a small amount of foliar food and the bacteria. Would that be detrimental?
Potentially. I could see the bacteria consuming food
The
that
food.
may or may not be a bad thing. If you want the bacteria, you put them in there in the first place, but they may change the balance, or
Right. That was my
Yeah, I would add them separately.
Okay.
But you
So
can definitely
2 IBC totes.
right? or either that, or just you can add the bacteria to the tote, but don't store it that way. Like if
Okay, so just add it per dose. Yeah. See, I was trying to get more automated because I have a lot going on. So I was trying to find a way to get everything on one system. But if I can't, I can't. It'd still be easier than backpack spraying.
Yeah. The backpack spraying is probably the best way to apply these bacteria, though. And
It
even
is. Yeah. Because I can see it. Everything's getting it, and like that's why every time I've set up automation before, my plants kind of like degrade a bit in quality, because with the backpack sprayer or me physically going over, I'm like, my OCD or anal retentiveness is like every little leaf,
trying to get under the underside, and
yeah.
I'll send
Especially
you
on the crypts.
I'll send you a really cool paper too, actually, for using these bacteria for foliar feeding.
Awesome.
You might find that really interesting.
Definitely, please. But yeah, send me that link of the study that you said in private chat really quickly, and I'll post it up.
Okay. Chat with so it looks like chat with everyone in the studio. Does that work?
I believe so. I think that goes to private chat.
Let me see if I can find it. If I get back in here, then I can, because I already had it copied, so I could paste it on the old computer I was using, right?
Yeah.
So if I enter the studio, I can do that. Looks that way. Cool.
Oh, there we are again. I'm just joking.
That's pretty cool. How's that?
There we go. I see it.
Right on. Cool. And I'll find the other one. I'll just send that one to you in
perfect
in Messenger. But it's good talking to you guys all the night.
Yeah, no, I really appreciate your time, man. That was a great conversation. It provided me, like I said, it gave me that little reference now back to talk to people about the importance of, a stable aquarium. It goes more than just your fertilizer, more than just your lights, and more than just your CO2. Proper horticultural care and setting up a good ecosystem is key. Beyond that is just icing on the cake, So, but I appreciate your time, man. Thank you.
Likewise. We'll stay in touch. Have a good one.
Bye.
See ya.
All right, so guys, little catch up now. Grant, how are you doing?
Wait for your answer. It's the end of the stream. I don't know.
Actually, it's not quite the end. Brian, what do you got going on?
I got my setup going. I'm still gonna play around with that this week.
Okay. Can we get a quick look at it really quickly?
Yeah.
Because you did change some stuff.
Yeah.
I know Grant wants to go to bed, but I'm gonna keep him up a little bit longer. So there we go. Nice. Now, I take it your soil would be at different heights throughout the aquarium, obviously? Yeah. Okay. Yeah. I can kind of see where it would be higher and where it would be lower.
And I'm going to build up over here more so I can do some more depth work.
Okay. Wow. There we are. Nice. Nice. So, Grant.
And now I'm going to break the tank with my chair.
You're going to make me commit to this, aren't you? I can already tell you want to do it.
So there was a couple things that went into play. I wasn't going to do it unless Eddie was going to go, and Eddie's going to go so he can watch my booth Saturday. So I kind of have like availability now.
Yeah. Man.
I also would like the advantage of going around the round robin before we have to do it in Chicago.
I hear that, and that's like the only reason I want to do it. But at the same time, it's like creative juice that I could use in Chicago. Because I'm like I don't know how to put it. Like one of those rechargeable batteries from the 80s where you press a button on it and it says how charged it is.
So
I'm just worried if I go to Dallas and try to get creative, I'm not going to be able to be creative anymore.
Here's what I'm going to do at Dallas. I'm going to take a look at the hardscape options, and then I'm going to go and try and track down Sean and be like, are these going to be, similar hardscape options for, Chicago? I can't imagine there being much difference, from one to another. And I'm going to pick 2 scapes that I really want to do in Dallas, and I'm going to execute the second better option, for Dallas, and then I'll try and persuade us to do the better option for Chicago. But like as a team effort, Brian can go, let's do this, and you can go, you know what I mean? So I don't know. As right now, it's just a collaboration between me and Shelby. If you want to join in, but
I don't know, man. I guess you can put me on to help. I don't know how much I'll contribute, but at the same time, I may like overly contribute or get too into it. And I don't want to be pushy because this is something that you and Shelby have started to do. And usually I try to like take control and have design say and stuff like that. And I don't want to do that here. So I'd rather hold that till Chicago.
It's more along the lines that it is kind of like time crunching too. So if Shelby has to do something with the shrimp contest, then it would be nice to have somebody else's hand there. I don't know.
What he's saying is he wants you to smash some stones for him.
I'm just waiting for him to ask me to join. I'm just joking. I'm just joking. Yes, whatever. Let's do it. Let's try to win Dallas.
Unless you were talking to Kevin and Kevin was going to join and you guys wanted to partner up, but I want to get as much of an advantage going into Chicago as possible.
No, I wasn't talking to anybody about competing. I have tanks I got to do at Aquashella for companies and stuff, so I'm going to be a little predisposed, but I will probably have most of that done Friday.
Right.
I have to have it done Friday.
Are you just doing OASI, or are you doing?
I gotta clarify all that because I started some trouble, I guess, by talking to JBJ. Because Owase, I guess, is direct competitors with JBJ, and I didn't realize that. So I don't want to burn my Owase bridge, but at the same time, you can only live off reputation cookies or exposure cookies for so long. JBJ is actually willing to pay me money. So there's a little difference there, but at the same time, I love Owase. They've done a lot for me and continue to do a lot for me. So I'm in a pickle. I don't know what I'm going to do yet, but I gotta let Joe know sooner than later, and I gotta let Ashley and Jen know sooner than later. Otherwise, I won't be doing anything.
Oh yeah. Yeah, I got to figure out if I'm doing something for Carib C or not. They said definitely, like, let's do something at the show. And I was like, I don't know if I'll have time to set something up at the show because, like, they usually set their booth up, like, last minute, last day. Like, they've done it so many times. They just know that they only need an hour to set up their whole booth. But then that gives me 3 or 4 hours to set up the scape because I put it on the table and I don't know where their stuff's going and stuff like that. So I'm like, it really worked out for me last time where I just dropped my award-winning tank from the state fair onto their table. So I was like, hey, I can do something similar to that. We have a 25-gallon and a 10-gallon rimless aqua top setup that we got from Aquatic in Tennessee.
Yeah.
And me and Shelby both have really nice Daenerys Scapers tanks, and they're JBJ friendly. So I'm like, buy me a brand new Scapers tank. I'll just bring one already scaped, and then I'll take the other one home and scape it for another show or something. So I don't know.
Yeah, I got to do 2 JBJ
Yeah, I know. I don't like that style though.
dinner lay. I got to do 2 rimless dinner lays, 10-gallon, and then I got to do one of their 25-gallon all-in-ones for JBJ. I don't even know what Awase had for me. From what I was told, it's like this really long bookshelf tank. I don't know if you remember the one that Melanie did, but it's that really, really long, narrow bookshelf tank. So like that would be challenging, and I could probably have some fun with it, but I don't know. We'll see. I wish I could just do both. It's so stupid. Like, why can't I just be an independently contracted artist? It's not like I don't have a contract signed with either of them. So I mean, come on,
Free agent.
you know, it makes no sense. That's like telling Michelangelo he can't paint the ceiling of somewhere else because he painted the ceiling there. It's like, it's silly, but I guess it is what it is. That's business. But yeah, let's do the competition. Let's see how it goes, and that way we can at least get a taste and see what life is like in the world of pre-wrapped hardscape, because that's what it is.
Cholo.
Wrapped.
We'll figure it out.
Yes.
You all know Cholo once.
So, all right. What else do we have to talk about here, guys? Are we done?
I think we're done.
I think we're
I'm
done.
gonna go Cuban
Yeah.
tree frog hunting.
Why? So you can bring him to Grant's house?
Nah, so I can. Well, my neighbor found a BB gun on the side of the road, and he's like, "You want it?" And I said, "Yeah."
I was probably used to kill somebody. Now you got a murder weapon.
Not a BB gun.
It's like
You never
it's
know
like a
these days, man.
spring, one pump spring loaded BB gun. Like 9 times out of 10, the frog is like, "That's all you got."
All right. Well, I appreciate everybody for tuning in. There is something that I need to talk about really quickly. I got 1,000 subscribers apparently. I was just looking back at the chat, so that's awesome. I really appreciate it, guys. That's huge. So what I'm going to do, because I talked about the giveaway, and I'm doing this at the end of the episode, the end of the 2.5-hour episode on purpose. Maybe so people watch the whole thing. Probably not though. But I'm going to, on episode 10, the next even episode that'll be on my channel, we're going to facilitate this giveaway on this thing here. I'm going to learn how to do the thing that Shelby taught me, where I can actually have the StreamYard pick a winner type stuff. So it's going to be interesting, and I have to do it 4 times, so I'll likely mess up, and it'll happen a couple of times. But you got to make sure you're logged in, from what I understand, in order to be counted by it, right?
You just got to be a subscriber, and then you just hit the hashtag.
All right, so we'll figure that out, and then we'll get that going. But that's how we're going to do it. So I really appreciate it, guys. Anything that comes from the future of my channel is going to be put directly back into the hobby, either through the Plant Library Conservation Project or the Florida Aquatic Plant Club. Aquascape is a business entity that I use to feed myself and have fun, not to take money from YouTube. Like, if those that wonder why my YouTube's Chance Kramer and not Aquascape, it's because Aquascape is for everybody. It's not anything that I want tied to just me. You'll start to see more people pop up using their name from Aquascape, like Eric Smithson, Mark will be coming on. It's basically a big team of people looking to do something positive in the hobby. So that'll probably be its own channel in itself too later on down the road. So, but I appreciate it, guys, and thank you, Scape Team, again for coming on. You guys are awesome. And Dallas, look out, because Grant and I are going to come and glue together a bunch
Shelby.
of stuff. And Shelby and everybody,
man.
you know what I mean? I'm not in, I can't, there can't be offense to when I talk. If I leave your name
guys
out,
are
it's not a big
together.
deal.
Let's just start calling Grant Shelby, and
You
then
know what I mean?
we're good. We'll always forget Grant.
I'm just gonna start forgetting Grant then and see how that
There
works.
you go. That's better.
Shelby and I are gonna kick
Mhm.
Dallas's ass then. How does that make you feel, Grant?
It feels better.
Good. Okay. Well, I appreciate all of you, and
Yeah.
I hope you guys have a good night. All right.
Later, everyone.
Bye, everybody.